Hello, and thank you for joining the Peach Education Conference as part of the 2021 Virtual Southeast Regional Fruit and Vegetable Conference. I am Andy Rollins, County Extension Agent for Clemson University, and I will be the moderator for this presentation. Pesticide credits, CCA credits, are available for most presentations offered during the live Southeast Regional Conference. Please check the Pesticide CEU Guide for a list of approved presentations and participating states. The Pesticide CEU Guide is located in the Event Resources tab under the Media Player. The guide is also available at the Resource Center located on the main menu of the conference platform. Please note that pesticide credits will only be available for registered attendees and only during the live conference. Credits will not be available for on-demand viewing. There is a three-step process for receiving pesticide and CCA credits. First, go to the audience chat box located on the left side of your screen and type your first name, your last name, and the states for which you are requesting credits. Again, the audience chat box is located on the left side of your screen. Type your first name, your last name, and the states you're requesting credits from. You will need to do this for every presentation to request the pesticide credits. The second step is to sign out at the end of each presentation. In order to sign out, it's the same procedure. You go to the audience chat box, you type your first name, your last name, and the states you want credits from, and then you will be reminded to sign out at the end of the presentation. The third step is to complete the pesticide CEU registration. You only have to do this one time to complete your registration, and it's not required for every presentation. To access the Pesticide CEU registration web link, open the Pesticide CEU Guide on the Event Resources tab located under the Media Player. The Pesticide CEU registration web link is located on the front cover of the guide. This presentation is being pre-recorded in order to reduce the technical difficulties. We will be answering questions live at the end of the presentation. You can submit a question at any time during this presentation by typing your question into the question box and pressing send. Don't forget to thank the 2021 Southeast Regional sponsors and exhibitors by visiting the virtual trade show and the featured product pavilion. They're the reason that we're able to have this. Lastly, don't forget to join us each morning for coffee chats beginning at 8:15 a.m. and also in the evening for networking beginning at 5 p.m. Now I'd like to introduce Dr. Hehe Wang with Clemson University. She will be presenting on chemical and biological control of peach bacterial spot. Dr. Wang has her PhD from The Ohio State University and has worked on her postdoc at Clemson University. She has done extensive research on bacterial diseases of strawberry and peach, and also has a strong background in soil diseases, Phytophthora and Fusarium. I am greatly looking forward to hearing from what Hehe has to teach us. Hehe?
Okay. Thanks, Andy, for the introduction. Today, I'm going to talk about chemical and biological control of peach bacterial spot. So 2020 has been a bad year for most of the people, and the actual rainfall this year also made bacterial spot really bad in the South Carolina peach orchards. In July, you can see nearly 100% of fruit infection and defoliation in some of the peach orchards. So here, I would like to use some pictures I took this year to help give a quick introduction about this disease and how the disease symptoms progress over time. So bacterial spot is also called bacteriolosis, that it's caused by the bacterium pathogen Xanthomonas arboricola pathophruni, in short ZAP. So these bacteria got splashed around in the peach orchards, mainly through the splashing rain or wind-blown water droplets. In the early spring, the bacteria that overwintered on peach trees will start growing and form canker on the young twigs, so mostly the one-year-old twigs. And then these spring canker serve as one of the major inoculum sources to cause leaf and fruit infections. And the young lesions on fruit look water-soaked. So when you hold the leaves against light, you can see that these lesions will look lighter green and translucent compared to the surrounding tissues. And when the lesions get older, they turn brown and they can merge together to form bigger patches. Usually you can see them by the yellow leaf tip. And eventually these infected leaves will fall off, and the early defoliation that caused by bacterial spot will make the tree weaker and could impact the winter hardiness. And sometimes it can also reduce the fruit size and cause the cracking on fruit. On the other hand, the bacterial spot on fruit start as little speckles in the late spring, and these lesions can grow into deep crater lesions later on in the season. Then the bacteria got the splash around on the peach trees and will infect the twigs and buds and leaf scars again and overwinter in this tissue to cause another round of infection in the following year. So bacterial spot could have very similar look to other leaf spots, like those caused by the chemical burns. So sometimes it's very difficult to tell them apart because all of them look like brownish, and they could, when the dead leaf tissue fall, they can form shot holes, and they can have like yellow halos around the lesions too. So here are some tips that can help you differentiate bacteria spot from the chemical burns. That the number one tip is the shape of bacterial spot is angular. That the bacterial spot is confined by the small leaf veins. Even when the lesions merge together, they still have an angular edge. While the chemical burns, they mostly have a round shape or irregular shape. And also a lot of times you can see the bacterial spot by the yellow leaf tip or along the midrib, or sometimes they got scattered randomly on the leaf surface. While for chemical burns, they mostly got scattered randomly on the leaf surface and they can gather along the leaf tip. But then you don't normally see them along the midrib. So here are the factors that can contribute to bacterial spot. The number one factor is the warm and humid climate, especially wet springs, which is the traditional climate in the southeast. And fruits are most susceptible between shock split to pit hardening. After pit hardening, the fruit becomes less susceptible, but if you have a lot of infected leaves and warm and humid climate, the fruit infection will continue along the season. And the trees growing in sandy soil is more susceptible to bacterial spot than the trees growing in clay soil, partly because the wind-blown sand could cause wounds on plant tissues, which will increase the bacterial infection. And the stress trees from other disease pressures or abiotic stress and the trees with poor vigor are also more susceptible to bacterial spot. For disease management, it's always easiest to use disease-resistant or tolerant cultivars. However, the current tolerant peach cultivars can still show symptoms at high disease pressure, and they do not have the most desirable fruit traits. The cultural practice that can help with disease management include planting ground covers and windbreakers to reduce the strong wind-blown sand, but still allow the air movement through the orchard. And also sanitize your pruning tools as often as possible and during the pruning. And if possible, prune off those young one-year-old twigs with spring cankers. And also minimize other tree stresses and keep a good tree vigor. Currently, the most common disease management practices are still relying on the season-long application of copper and oxytetracycline products. But these chemical controls also have limited efficacy, that oxytetracycline can break down in 2 days in the sunny days, and the copper products could cause phytotoxicity, which will also contribute to early defoliation and impact the tree performance. So on top of these limited efficacy, we also found oxytetracycline resistant and copper tolerant bacteria in South Carolina peach orchards in the past 4 years. And this table shows you the percentage of bacteria with different chemical sensitivity level. As you can see, the oxytetracycline resistant bacteria and the high copper tolerant bacteria are relatively rare, but they were found in multiple farms in different years. But for the low copper tolerant bacteria, there was a big jump this year that almost all the bacteria we found have a low level of copper tolerance. And these level of chemical resistance or tolerance were determined in our lab assay. So in order to evaluate their true impact on the spray programs, we did some greenhouse trials on peach trees. So before we look at the data from our greenhouse trial, let's take a quick review of the copper dosage from our spray guide. The copper dosage can be as high as over 2 pounds metallic copper per acre during dormancy, or as low as 0.03 metallic copper per acre for the summer cover spray. So during the growing season, the copper dosage keeps decreasing, and basically after the petal fall, the dosage will stay very low, like a small, less than 0.5 pounds metallic copper per acre to prevent or limit the phytotoxicities. So in order to evaluate the impact of the copper tolerance bacteria on our disease control, we include 3 different dosages of copper in our greenhouse trials to represent the low, medium, and high dosage of copper in the spray guide. So on this graph, you see the gray bars are the amount of disease caused by on the peach leaves without any chemical treatment. And as you can see on this graph, when the copper dosage are lower than 0.5 pounds metallic copper per acre, it was only able to effectively control the sensitive bacteria, but it cannot reduce disease for the copper tolerant bacteria. For the high rate of copper, which is 2 pounds metallic copper per acre, it was able to control all the bacterial phenotypes, but the efficacy, control efficacy was lower for the copper tolerant bacteria compared to the sensitive bacteria. So here is another picture showing you the disease severity on the leaves from our greenhouse trial. It basically shows the same trend as what we discussed in the previous graph. And here's another 2 summary table showing you that the copper tolerant bacteria could impact our ability to control bacterial spot. That after the copper treatment, the copper tolerant bacteria still cause significantly higher amount of disease compared to the sensitive bacteria. So after copper, let's take a look at the oxytetracycline resistance. As we expected in our greenhouse trials, the fire line was able to effectively control the sensitive bacteria. That if you can see on these leaves sprayed with fire line, there is not much disease present. But when the fire line lost effect when the oxytetracycline resistant bacteria are present. As you can see that after fire line, the leaves still develop about the same amount of lesions as the no spray controls. And another thing that's significant from our finding is that all the oxytetracycline resistant bacteria are also resistant to a high level of streptomycin, although streptomycin is not currently used in the peach orchards. So because of the emergence of these chemical resistance and tolerance in South Carolina peach orchards, we started to evaluate new chemical control options. So these 4 are the chemicals we evaluated, and they have shown like success in to control bacterial spot in other crops. So zincicide has shown success on citrus, and it totally inhibits all bacterial growth in our lab assays. And nanomagnesium oxide and aminothiodiazole have shown promise on tomato and strawberry, but they did not perform that well in our lab assays. Mancozeb and pancozeb, they are not new, but they are not registered on peach. And combination of mancozeb with copper showed the best control effect for bacterial spot on almonds, which is caused by the exact same bacterial pathogen on peach. And so we include mancozeb in our lab assay, and it did show a perfect inhibition of our bacterial growth. So after the lab assay, we include some of these chemicals in our greenhouse trials. So we did another 2 greenhouse trials and include 1,000 ppm of zincicide, 1,000 ppm of nanomagnesium oxide, the regular rate of fireline, and the low-medium rate of coside at 0.3 pounds metallic copper per acre. So a little bit different from what we found in the lab, the zincicide was only able to effectively control the sensitive bacteria in our greenhouse trial. In fact, none of the chemicals was able to control our oxytetracycline resistant and copper tolerant bacteria in our greenhouse. And in addition to zincicide, fireline was the only chemical that was able to significantly reduce disease from the sensitive bacteria. But the reduced performance of zincicide in plants compared to in the lab assays are also commonly seen in the other crop systems. But the good thing is this zincicide at least can provide some control for the sensitive bacteria. And with this high dose of zincicide, it causes minimum amount of phytotoxicity, which is much better than the phytotoxicity caused by the low medium rate of copper. So summarize from our research finding our chemical controls, it's the low copper tolerance becoming more prevalent in South Carolina, which could affect the copper efficacy on disease control, especially for the low dosages after petal fall. And the oxytetracycline streptomycin resistance luckily still occurred in low frequency, but they were found in different farms in multiple years. And the new chemicals that had success on controlling bacterial spot of other crops did not perform that well on peach. And among them, the zincicide was the only one that showed potential on controlling sensitive bacteria on peach. And here is a project that we'll start working on next year on peach bacterial spot. And in this project funded by USDA NIFA, we're going to continue monitoring the oxytet resistant and copper tolerance in South Carolina and Georgia peach orchards and evaluate their impact on the current chemical control programs. And we'll also evaluate the impact of the current chemical control programs on the emergence of these resistance and tolerance. And we'll identify the mechanisms behind these resistance and tolerance and evaluate more chemicals for improved control of bacterial spot. And at the same time, we'll also study whether these chemical resistant tolerant bacteria can overwinter on peach trees and contribute to the infection of next growing season. And we'll study the environmental factors that could affect their overwinter survival on peach trees. And then we'll develop a model based on these environmental factors to predict inoculum levels at the beginning of the growing season. And we'll keep everyone posted on our results in the coming years. So now let's switch gear and talk about the biological control options for peach bacterial spot. So biological control or biocontrol, the traditional definition is to use a living organism to suppress pests. The commercial products that have been labeled for peach bacterial spot include double nickel, Lifeguard, Serenade, and Serafel. And they have 3 common modes of actions. The first one is they can directly antagonize pathogen with secreting antimicrobial metabolites, or they can compete with pathogen for food and space on plant surface, or they can induce plant defense. So Lifeguard is an example of a biocontrol product that induces plant defense, while double nickel, Serenade, and Serafel all have the first 2 modes of action. The biocontrol products normally do not cause phytotoxicity, and they are advertised to be used as more sustainable options to rotate with copper under low to moderate disease pressures in the growing season. So the current spray programs for peach bacterial spot mainly focus on from late dormancy to the end of growing season. But the late fall is also a critical period for the bacteria to cause new infections on leaf scars, twigs, and buds, and overwinter as inoculum source for the next growing season. And the dormant application of copper and copper mancozeb was found to significantly reduce bacterial spots in almond, which again is caused by the exact same pathogen on peach, in seasons with high rainfall in California. So seasons with high rainfall in California basically is the same normal seasons in the Southeast. So we think the fall application of biocontrol could protect the trees from the new infections at that time and reduce the inoculum source levels for the next growing season. And the biocontrol agents may work better in fall than in the growing season because the bacterial pathogen level in late fall will be lower than in the growing season. And the mild fall weather in the Southeast and the absence of other pesticide applications could favor the survival of these living organisms on peach trees and increasing their chance of success to fight pathogens. And we conducted a field trial last year in the local peach orchards. We did the 4 sprays before forecasted rain event, and we found the spray of biocontrol products and or copper all associated with the less spring canker in early 2020. And this year we're repeating this trial. In addition to the commercial products, we also found 14 local biocontrol strains from South Carolina fields, and they show comparable or higher degree of antagonism against the bacterial pathogen than the double nickel and serenade in our lab assays. And these bacteria may work better in the local environment than the commercial products, and we will evaluate them in our future spray trials. So the take-home message from the presentation today is the biocontrol sprays during late fall have shown promise in reducing inoculum for the subsequent growing season. We are still repeating our trials, and these, if they continue providing like a positive result, these can be a potential option for both organic and conventional growers to improve disease control in the future. And the low copper tolerance is becoming more prevalent in South Carolina, which could impact the efficacy of low copper dosages during the growing season. And we will evaluate the alternation of copper with other products such as biocontrol agents, zinc, and antibiotics after pedofall in our field trials. And oxytetracycline streptomycin resistance we found luckily still occurred in low frequency, but it has been found in multiple farms in different years. And we'll keep monitoring these chemical resistant tolerance in the southeastern peach orchards, evaluating their impact and developing new strategies for improved disease control. With that, I would like to thank all the people who help out in this research, especially Dr. Guido Schnabel and Brody Cox. And Brody Cox is the student who worked on copper tolerance, and the extension agents Sarah Scott and Andy Rollins, who help us out in our research project, and all the people in my lab who helps out, and all the growers who give us a lot of support in our research projects. And our research are funded by South Carolina Peach Council, South Carolina Department of Agriculture, and the USDA NIFA. Thank you very much, and I'm ready for any questions.